Hello there everyone and welcome to this Biology Mind A-Level tutorial on cell division by meiosis. So, in today's tutorial we're going to have a really good look at the nitty-gritty of meiosis. Now in our previous tutorial we had a very general overlook of what meiosis is and why it's really important, and we had a look at the sort of cells that it makes and discussed how it introduces variation into populations as well. So if you haven't watched that video already, I'd really recommend you go back and just watch that. It's not very long. And in this tutorial, we're going to focus a lot more on what is actually happening in the process of meiosis. So let's kick off. So let's, throughout this tutorial, think about what are the main differences between mitosis and meiosis. So we'll see it as we go through and maybe make a list of little differences that you notice and that I point out, because it's really good to really understand how these two processes differ. So let's start off with the first difference. So meiosis is essentially 2 rounds of mitosis, if you want to simplify it in your head, but with some very important differences. So you start with one diploid cell in meiosis. So this parent cell is diploid. And normal body cells have the diploid number of chromosomes. So we refer to that as 2n. So normal body cells have 2n chromosomes. We know there are 23 pairs of chromosomes. So normal body cells have 46 chromosomes. And when I say diploid, all I mean is that each cell contains 2 of each chromosome, one from mum and one from dad. Now, gametes, which are what are formed in the process of meiosis, so these 4 cells in this column here are gametes, they are haploid. So this means that they have a haploid number of chromosomes, which is actually half the number of the diploid. So 2n here divided by 2 is n. So gametes contain half the number of chromosomes compared to diploid cells. So remember I said with diploid cells, they are called diploid because each cell contains 2 of each chromosome, one from mum and one from dad. Well, in gametes, which are haploid cells, there's only 1 copy of each chromosome. And you can see that here. So, for example, the shorter chromosome, there's only 1 copy of the shorter chromosome, whereas in the diploid cell, there's 2 copies of the shorter chromosome, just as an example. Okay, so gametes are the sperm cells in males and they are the egg cells in females. And they join together at fertilization to form a zygote, which we met in the previous tutorial. And the zygote, remember, is formed from that sperm cell, which has n number of chromosomes, plus our egg cell to form the zygote. So overall, we now have the full number of chromosomes within our zygote. So this is why meiosis is so important. It's important in making sure that gametes have half the number of chromosomes compared to a normal body cell, because that means that when the gametes join together, they then have the normal diploid number of chromosomes as a zygote. Okay, so let's move on from here. So this is the first difference I want you to notice, which is that meiosis makes haploid cells from diploid cells, whereas mitosis makes diploid cells from diploid cells. Okay, so let's move on to have a really good overview of what meiosis is. So this is a very zoomed out version of meiosis, and I'm just going to talk through this diagram really slowly. So you can see here there's 2 stages of meiosis. There's Meiosis I, which is division number 1, and there's Meiosis II, which is division number 2. And before all of this, there's interphase. So interphase is part of the cell cycle. It's not specifically part of meiosis. So it comes before meiosis starts. And in interphase, what we get is we get a copying of the DNA. So the DNA unravels and it replicates that there are 2 copies of each chromosome called chromatids. So for example, let's have a look at this. So This is chromosome number 1, of which we have a maternal chromosome number 1 and a paternal chromosome number 1. So together we call those our homologous chromosomes. Remember, there are homologous chromosomes. And in interphase, each one out of these homologous chromosomes makes a copy of themselves. So you now can see that there's 2 of M1 here and 2 of P1 here. And one of these chromatids and the second of these chromatids, they're referred to as sister chromatids. So a little bit confusing to get your head around. But if you just remember that there are now 2 copies of each chromosome called chromatids, and together we call them sister chromatids. Okay, so these are our homologous chromosomes. They make a copy of themselves in interphase, and those 2 copies together that are joined together in the middle at the centromere, that's your sister chromatids. So I'll write that here, sister chromatids, which we'll talk a little bit more about a bit later. Okay, so in interphase we get the copying of our genetic material. Then Meiosis I starts, which is our process of our first division. And in Meiosis I, our chromosomes arrange themselves into homologous pairs. So we've met homologous chromosomes here. Now they've duplicated, but together these 2 are homologous pairs because they're both the same chromosome. They're both chromosome number 2. It's just that this is mum's chromosome number 2 duplicated and this is dad's chromosome duplicated, and the same for chromosome number 1. So these are now, this set is one homologous chromosome and this set is the second homologous chromosome. So the chromosomes arrange themselves into these homologous pairs, and these homologous pairs are then separated in Meiosis I, which halves the chromosome number. So when I say they are separated, have a look, for example, with this homologous pair, M2 and P2 are separated in the daughter nuclei. So Meiosis I is all about separating the homologous chromosomes. That's what I want you to jot down, and that's what I want you to remember. So let's think about the chromosome number now. So here we had a diploid cell, 2N, and in interphase we've doubled that number because remember we replicated our DNA. We've now got 2 copies of each in the homologous chromosome. And then in Meiosis I, we split this apart again. So we are now on this because we've got 2 of each type of chromosome. Because we've divided this now by 2. Oops, sorry, that wasn't meant to be a 2 there. 2 times n. We've now got 2 of each type of chromosomes. We've got 2 sister chromatids of chromosome number 2. But chromosome number 2 itself is inherited from the maternal side. Whereas here, we've also got 2 sister chromatids of chromosome 2, but from the paternal side. Okay, and then we get the second division, which is Meiosis II. And this is where the pairs of sister chromatids that make up each chromosome are separated. So, for example, let's look at P2 here. You can see that this sister chromatid of P2 has gone to this nuclei, and this sister chromatid of P2 has gone to this nuclei. So Meiosis II is sister chromatids from each chromosome are separated. So we had interphase, where we had the duplication of our DNA. We had Meiosis I, where we know homologous chromosomes are separated. And then we had Meiosis II, where sister chromatids from each chromosome are separated. Now, what is the result? We have 4 haploid cells over here. These are our gametes that are genetically different from each other, and we'll talk a bit about how they're genetically different from each other later on. Okay, so something I want you to remember is also we've only shown 2 chromosomes in these diagrams, so chromosome 1 and chromosome 2, but in all of these nuclei at any time, there's at least 1 copy of everywhere from chromosome 1 to chromosome 23. So there's always the full set of chromosomes. It's just that the numbers of each chromosome changes with each division. Okay, so we've gone from our diploid parent cell to our haploid parent cell. Our haploid daughter cell. Now let's have a think about our different stages of meiosis. We had our interphase, our Meiosis I, our Meiosis II. We're going to break that down a little bit more. So this is a bit of extra detail now. So before Meiosis I told you that interphase occurs, so that's in the cell cycle, and this is DNA replication and growth of the cell. So interphase consists of two phases, the S phase, where DNA is replicated, which you can see here. We've now got two of each in the homologous chromosome replicated. And we also have the G phase, which stands for the growth phase. So growth phase 1 and growth phase 2, where new proteins and organelles are made, resulting in our cell growing a little bit. Now, it's important that new proteins and organelles are made because remember, at the end of the day, in meiosis, we're splitting up the cell into 4 daughter cells. Those daughter cells need their own proteins and organelles in them. So we've got to grow those new proteins. We've got to develop those new proteins and organelles at some point. So that makes sense that it all happens in the interphase stage before any division occurs. Then we enter Meiosis I. So we know that Meiosis I results in the production of 2 diploid daughter cells. Now each of the phases of Meiosis I are the same as normal mitosis. So we have prophase 1, metaphase 1, anaphase 1, and telophase 1. So in prophase 1, this nuclear envelope that surrounds our chromosomes breaks down. Okay? Meanwhile, our homologous chromosomes are still in our nucleus, but the nuclear envelope is breaking down. And we have these things that form at the poles of the cell, at the ends of the cell. And they are called centrioles, and they branch out these spindle fibres from them. And these spindle fibres grow towards the homologous chromosomes. And you can see in metaphase 1, the spindle fibres have attached to the centromeres, so the central bits, of the chromosomes. And the homologous chromosomes have lined up in pairs, so chromosome 1 with chromosome 1, chromosome 2 with chromosome 2, so they've lined up in pairs along the equator of the cell. And the spindle fibres have attached. Then in anaphase 1, the spindle fibres contract and pull the homologous chromosomes apart to the opposite ends of the cell. And in telophase 1, the nuclear envelope reforms around those separated chromosomes. And remember I said in Meiosis I we're separating those homologous chromosomes. So one in the pair goes to one nuclei and the other in the pair goes to the other nuclei. And then we enter Meiosis II. So the fact that we've now got prophase 2 starting means Meiosis II is about to start. Okay, so before I talk through Meiosis II, I'd just like to go back to Meiosis I and talk a little bit about what our end phase of Meiosis I is. So we get those 2 diploid daughter cells which have formed at the end of Meiosis I. Our homologous chromosomes have separated, okay? Okay, so I hope that's nice and clear what we've done in Meiosis I there. Then, in Meiosis II, we have our 2 diploid daughter cells that formed from Meiosis I, dividing to produce a total of 4 haploid daughter cells, each with a single copy of every chromosome. So we'll see that at the end of this diagram. But again, we go through prophase 2, then we go through metaphase 2, we go through anaphase 2, and then telophase 2, where the cells then divide by cytokinesis to produce the 4 haploid daughter cells. Now, all of these phases are the same as we've just discussed in this tutorial, so I'm not going to repeat myself. But it's very important for you to understand that at the end, we get this production of our 4 daughter cells, which each have the haploid number of chromosomes. Okay, so we've gone through those stages in meiosis now. And let's just have a quick recap of that before we go on to have a look at how genetic variation is introduced in meiosis. So, before meiosis I, we said interphase occurs, in which there is this DNA replication and growth of the cell. And interphase consists of two phases, the S phase, which is the DNA replication phase, and the growth phase, where I said that new proteins and organelles are made. Then we go through meiosis I. Meiosis I results in the production of two diploid daughter cells. And remember, our components of meiosis I are prophase I, metaphase I, anaphase I, and the last one is telophase I. And meiosis I, remember, pulls those homologous chromosomes apart. Now, we've been through meiosis I, and we enter meiosis II. Now, before we go on to just recap meiosis II, I want to draw upon something that happens in meiosis I, or specifically in prophase I. So a process called crossing over occurs in meiosis I, and it occurs in prophase I specifically. Now, crossing over does not occur in mitosis, so this is one of the key differences between mitosis and meiosis. Now, crossing over is when the homologous pairs of chromosomes come together and they pair up, as you can see here. Then what happens is the chromatids twist around each other, so you can see they're slightly overlapping here. Chromatids twist around each other and bits of chromatids swap over. Now remember, when I've talked about chromosomes in the past, so let's say this is one in the homologous pair and this is the second in the homologous pair. At any point here on this chromosome, this is a loci, so a specific region where a gene is located on the chromosome. Now, in crossing over, because the homologous chromosomes are lined up in line, when they exchange bits of their chromatids, they're exchanging bits at the same loci, so at the same gene. So really what they're doing is they are switching up their combinations of alleles. So if they're switching up that same bit of DNA on that chromosome, the resultant chromatid that results at the end of crossing over still contains the same, so it still contains the same genes, but we now have a different combination of alleles. So this means that each of the 4 daughter cells formed from meiosis contain chromatids with different alleles. So this is one way in which meiosis produces cells that are genetically different. We get this introduction of genetic variation because of a process known as crossing over, where we're swapping positions on the chromatids at the same loci, so the same genes, but it means that we swap over different alleles. So overall, the daughter cells end up with a different combination of alleles. Now, the remainder of meiosis I is exactly the same as mitosis. The homologous chromosomes are separated from each other, and these are sorted into 2 diploid daughter cells. And then we enter meiosis II. So again, prophase II, metaphase II, anaphase II, telophase II, and then we get our 4 daughter cells, which are haploid being formed. Now, something that I want to draw upon here is that we get, in meiosis II, we get our 2 daughter cells that were formed from meiosis I, dividing to produce 4 haploid daughter cells, each with a single copy of every chromosome. Now, let's go into those little details of meiosis II again. So if the nuclear envelope was remade after the end of meiosis I, so at telophase I, it will break down again because we need to release those chromosomes again for the spindle fibres to get to them. The chromosomes condense and spindles are made at the centrioles here. Again, as what happens in metaphase 1, in metaphase 2, the chromosomes line up in the centre of the cell, or we sometimes call it the equator of the cell, and spindle fibres bind to the centromeres, so the centre bits of the chromosomes. And the chromatids of each chromosome are independently assorted. So all this means is that within the homologous pairs, chromosomes are randomly organised on either side of the equator. So this means that it's completely due to chance which side of the equator the maternal or the paternal chromosomes are pulled towards. So we call this process independent assortment, or you sometimes might have heard of it as independent segregation. So we know that all our cells have a combination of chromosomes from our parents, so half from our mum and half from our dad. But ultimately in our gametes, we have different combinations of those maternal and paternal chromosomes that go into each of those gametes because of the process of independent assortment. And we'll talk more about that in our next tutorial. Now, the spindle fibres in anaphase contract exactly like in anaphase 1 and anaphase 2, they contract and they pull the chromatids to opposite ends of the cell. And again, because of that independent assortment of chromosomes, we're getting different. It's completely random whether this chromatid is pulled to this side of the cell or pulled to this side of the cell. And then in telophase 2, 2 nuclear envelopes will develop in each of those daughter cells to form 2 haploid nuclei. So remember, it's now got half the number of chromosomes. And the 2 cells divide by a process called cytokinesis to produce 4 haploid daughter cells. Now, what is cytokinesis? This is just when 2 cells divide, or when the cytoplasm of 2 cells divide to produce 4 haploid daughter cells. So this means that overall, we've gone from having our diploid cells to having our haploid daughter cells. So remember I said that our diploid cells contain 2n, the full numbers of chromosomes, so they've got 2 in each chromosome, whereas haploid cells have every single chromosome from number 1 to number 23, but only 1 in the pair, because remember our haploid gametes are going to join together to then form that 2n, where they have 2 in the pair of each chromosome. Okay, so we've covered meiosis in a lot of detail. Well done for following through till the end. I hope that made a whole lot of sense. Just remember, try and pick out those differences between mitosis and meiosis to help you really understand what's going on here. And also have a think about how meiosis leads to genetic variation in the gametes it produces. Remember, we had the crossing over and we had the independent assortment of chromosomes. And I want you to think really hard about how those two lead to those different combinations of genetic information being divided into those final cells. Remember, in crossing over, we're mixing up those alleles between mum and dad, so we're getting chromatids that contain different alleles ending up in our daughter cells. And in independent segregation, we're getting that independent movement of sister chromatids to opposite poles of the cell. Okay, well done for following through. I'll stop talking now and I'll see you for the next tutorial.